5, 4, 3, 2, 1, 0.
High and low, it's how I roll. A shooting star in a film noir. I'm floating around 10 feet on the ground. I'm floating around 10 feet on the ground. I am no average Joe. Be with me, and you will see more than you ever wanted to.
Hey there, welcome all. This is Dr. John Baranchok saying good afternoon, late or early morning to you guys, or what other times you may be listening, I guess, ultimately in the future, hopefully. This is Grasping Sasquatch, Stories and Science, and generally try and tell a story that combines or is related to the topic of the day. Lots of articles and stories on the topic of today, but we'll get into that in a few seconds in the show. I think I'm going to do ground rules here. If you got questions, please type them in capitals, and that's going to be more likely to get my attention, and we'll put them up on the screen and I'll try and answer them. Don't have all the answers. If I've got an opinion, I'll let you know, and there's not too much I don't have an opinion on, so we'll see what happens. Yeah, questions in all caps. Converse amongst yourself in lowercase letters, please. If you like what you hear or any insights you might get, please subscribe, give me a like, tell your friends. Just promote the show in whatever way you see fit, but subscribing would be awesome. That's the big help. Let's see. Who I'm going to say hello to tonight. I see a lot of my regulars here. Uncle Bones, hello, how are you? Kevin Harder, waiting. Welcome to the show. John Ayers, how are you, buddy? Got the comic relief for the evening. Who else? Charlotte J. Eric Sell. How are you, my friend? Eric's a bright fellow. What else do I gotta say, promotional wise? That's it for now, I guess. Enjoying this new time slot. Not feeling quite as rushed to get through my material. We'll see how that goes tonight. I can't put my finger on. In my past few shows, I was reading from Jeff Meldrum's book, Sasquatch Legend Meets Science, on the Patterson-Gimlin film, and Dr. Dr. Meldrum's kind of summary and some interview material was in there, and I'm trying to get back to that. and just continue reading about that. It's kind of interesting hearing the words of Bob Gimlin. Okay, so they went up there based on Bob Green's advice. Where are we here? Okay. This Blue Creek connection, I think, was lost for me, but in the process of this sighting, this gentleman, I just said his name unconsciously, told Patterson and Gimlin that he found tracks at Blue Creek. And somebody claimed that some of that movie from that was cut into part of the Patterson-Gimlin film. I don't know if that's factual or not. Here we go. Okay. Picking up with Bob Gimlin's experience, and I don't do a good Texas accent, but I'll try and throw some twang in it as I imagine he might. He said, "It wasn't looking directly at me, but I could see the face real good, and I could see the eyes." Bob Gimlin recalled describing their initial encounter with the creature. As for the details of its face, the face would have a flat-type nose, the lips. I can't really remember what the lips look like, except it did have lips, and we could see its teeth. The eyes were large eyes, but not big round eyes. The hair on its face was short. There wasn't a whole lot of hair around its cheeks, down alongside the face. The best I can remember, its face didn't have a whole lot of hair on it. The creature walked away with an uneasy motion, swinging its arms like a human. Gimlin, familiar with livestock, was especially impressed by the evident and dynamic musculature. Yes, I could see the muscles clearly, and that was one of the deciding factors, in my opinion, that this was no man in a suit. The thighs and buttocks and the arms and shoulders. You can see it move clearly underneath the hair. Regarding the creature's gender, he continued, well, it appeared to be a female, but I've never seen one. I'd never seen a track until that day, so I couldn't make any kind of statement whether it was a male or a female, but the film indicates it had mammary glands, so we assumed it was a female. Something that males had mammary glands. Since the tracks found earlier on Blue Creek Mountain were of 3 different sizes, Patterson and Gimlin had assumed there were a male, a female, and a younger one in the area. In retrospect, the 3 were probably a female and 2 offspring, perhaps even the female they encountered. The prospect of the possibility of 2 more enormous creatures being in the vicinity, perhaps even a larger male, was a bit disconcerting. Gimlin was careful to ensure his horse didn't bolt from the scene. He continued, It was moving away from me. That was about all that was in my mind at the time. that this creature was no threat to us. And oh yeah, I was trying to keep my horse under control because, I never had any idea what might happen, and I sure didn't want to be on foot. So I knew I could get back on my horse if I had to. If I had to shoot it and it didn't go down, I could get on my horse and I could get out of there. And Roger wouldn't have to fend for himself. I'm not a coward, but I'll be damned if would have fended for himself. I'm not a coward, but I'll be damned if it's going to stick around if the creature got violent. So I wasn't concentrated. I was concentrated on keeping my rifle in my hand and my horse under control. So just reading through that, the one thing that strikes me about that is that he had to consider the possibility of shooting it, and chose not to. The creature continued its course of retreat upstream, disappearing around a knoll that dropped down from the canyon wall. But Patterson, still on foot and without his rifle, was nonetheless uneasy about the situation, said Gimlin. Then when the creature did disappear up the little draw, why, I wanted to follow it. Of course, Roger didn't want to follow it because he was on foot and he didn't want to be left there. We thought there was a possibility that there were two others. We didn't know at the time whether that was the ones had made the tracks up above the scene or not. Roger was a little bit upset so he wanted to catch his horse and get some more film in the camera. It took quite a while to catch the horse and to catch the pack horse. Well, as we tied them up, then we rode up on pursuit of the creature. I think I'll end it there. Wow. I've never met Bob. Seems like quite a man. But not related to the topic, but I think interesting reading again. Sasquatch, A Legend Meets Science, Jeff Meldrum. When was this thing published? (2006) by Boom How Ventures Incorporated. Forward copyright by George B. Schaller. I know a lot of people who own this book, and they let it sit up on the shelf and collect dust. If you have a serious interest in Bigfoot and knowing what was available out there back in 2006, or mostly before then, probably, it's an awesome book if you have it to crack open. It figured heavily in the writing of my book. I can't tell you how often I cite it. That book in my own book. Michael Federer, he could pass for a Pennsylvania accent as well. That's where I'm from. I'm from Pennsylvania, Michael. Nice to see you here. Kevin Harder. Yeah, somebody said, oh, there it is. That's what I was shooting for. As a Canadian, that sounds like a Texas accent to me. Yeah, I don't know. I've lived down in the South long enough to know there's a difference between a Texas and a North Carolina accent. I think my accent's kind of favoring a North Carolina accent when I'm trying to do Bob. So, any questions about the reading? I hope you enjoyed that. Let's see, where are we going to go? Let's maybe try this screen. So tonight, we're going to tackle, I don't know about tackling. I don't think we're going to be breaking any new ground, but one of the things I love about doing this podcast is I try and update myself on the topics. Because my book was written 3 years ago at this point. And so just to kind of update myself, I touch base and always learn something new in preparation for these shows. So it is awesome. Part of the work is learning new things for me, especially about Bigfoot. The Ketchum DNA Bigfoot Study, Questions and Controversies. Well, we're talking Bigfoot here, but we're going to start with the structure of the human hair. Been a while since I've looked at this diagram, and what's interesting to me, I think the layers of skin are pretty cool. The epidermis is the top layer. I need to go big screen to be able to read this. There we go. The epidermis is the top layer. Dermis. So your epidermis is the surface of your skin. Yeah, but not only includes the surface of your skin, but the tissue underneath of it. Then you got the dermis and this subcutaneous. When you get a shot that goes into your skin, this is this area of tissue, the subcutaneous area of tissue is where they're trying to get that medication in there so it disperses well into your system. The hair shaft, hair follicle, hair root, hair bulb, hair papillae, and hair nerve. It does hurt to have your hair pulled out of your head. And it's interesting that, to me at least, that it is also not only, well, I guess it makes sense. If it's innervated, that nerve needs some nutrients, so it's going to have blood vessels there. So I guess you can get your hair pulled out to the point that it bleeds. Scalp wounds are very bloody anyways. I was in a I had a traumatic brain injury when I was 16 years old. And a scalp wound, I like watching UFC as well, if you've ever seen on UFC, when their scalp gets cut. It's not pretty. So now we're looking at human hair, and again, my source for everything of the first part of the show that we're going to be talking about hair, I have just taken, frankly, from the BFRO website. And so you can see, I'll give credit, and you can see the source on the screen. I'm interested in reading the description of Bigfoot to you and see if it jargons any questions for any of y'all guys. I don't know. Let's see. I've got to show the chat somehow in this big screen. I may be missing some questions. That's okay. We can go back and get them. This is an important picture to remember or to understand that these are all human hairs. On the left, you got a dark hair with typical fragmentary medulla. So the medulla figures largely, or conceptually at least importantly, in this study. The dark area that's broken up down the center of that dark hair shows you clearly the medulla, and that's a typical. And the reason the medulla is important is because that's where the DNA in the hair is. You know, they say that if you go get a haircut, they can know everything about you in terms of genetics or whatever. I don't know what all kinds of information they can get, but it's from that dark material. Same on the far right. So the right one is a right coarser hair is white with a continuous medulla. So this whole strip of dark hair, or in the center of that blonde hair, is a continuous medulla, where on the dark hair side there, the medulla is broken up. Again, what's important to understand about that is that that's where you get the genetics, that's where you get the DNA from in human hair. And then in the center there, you got the blonde that has no medulla at all. So I found that interesting, that you can have a blonde hair with with no medulla, right coarser hair. I don't know what coarser means. It looks like even a blonder of a hair to me, but I don't know that I'm right about that. Let me go down to the back screen so I'm not able to see if you guys got any questions as I'm going. Hold on, here we go. Turn up my mic. Hopefully that'll be better. I can just bear with me a second. I'll blast you out. Testing. You getting me? Any better? Hopefully. Doesn't seem to be doing well. Let me know if my voice gets any better while I try and go through some of these questions that I want to catch up on. I just tried to address the volume. Let me know if it's working or not. Turn up my mic. Wow. Got it all the way on. How about that? All right. Thanks to Kevin Harder. He rescued Techno Idiot. Thank you. I think we're now cooking on all channels. Kevin, you're my man. You tell me, give me a thumbs up. So let's go back since we got some time. Great. Thank you, Kevin. You're the man. Let's see if I can catch some of those questions. I don't like leaving questions hanging. Okay, so I saw somebody, where is that comment? Michael Fetterman, I just want to respond to your question if you don't mind. Hoping this can make me understand what went wrong with the Ketchum study. I thought we were so close. Towards the end of the show, Michael, I'm going to talk a little about that. But at some point, that conversation begins to just kind of be like a piling on Melba process, and I'm not here to pile on Melba. As it is, I'm going to be critical of her, but I don't know. The short answer, Michael, is that if you look at some papers on the genetics, if you look at some of the reviews done by real geneticists on her paper, which we'll be getting into, I think that's where the gap is between the geneticists and us, myself included in this category, the genetics deprived or genetics undereducated. I struggled through a couple papers in preparation for this show. And one gentleman I was talking to, I'm sorry I don't remember your name. I hope you're here tonight, though. You got to talk the language that you need to talk is DNAs. And if you don't understand DNAs, it's a hard thing to sift through, for it was for me at least. And but I didn't read any reviews of it done by a geneticist that were favorable. I'm sure Dr. Ketchum could put me on to some. I understand there were some favorable reviews. But I think to really understand the answer to your question, I think that's where you need to go. And at some point in the show, I will be putting up it's included in tonight's presentation, what you need to read, in my opinion. Let's see, any other questions? Thank you. I hope maybe you do come away with a better understanding of that. I'll try. I'll keep you in mind. John, volume, we're back to the volume. Glad we got that solved. Thank you, team. Paramedic. Why did she call it angel DNA? She didn't have any angels around to test against it. Tested against it. I'm not familiar with that. Paramedic. I don't know. I've never heard that she called it angel DNA, but that doesn't mean that it didn't happen. I'm not an expert on her. I'm not an expert on the DNA involved in her study. I don't understand it. It's a different language to me. But so if you want to care to elaborate on that, I'd be interested in learning more. Certainly feel free to share it, and we'll kind of take a couple detours through it. Or if anyone else is familiar with it being labeled or called angel DNA. That's a question I don't know the answer to. There'll be a lot of those. Any other questions? I think I'm caught up with the questions. So we were comparing the human DNA, or not human, well, where they get the DNA in humans to the Bigfoot hair. So let's see if we can go bigger on this. Yeah. So now we got two samples, purported samples of Bigfoot hair. You got dark hair on the left and reddish brown hair on the right. I would have called the reddish brown one the one on the left, but that's just darker. What's missing in both of those hairs? What's missing? Back to the humans. Back to DNA. Bigfoot. There's no medulla in there. In two of the three hairs in the humans, we had the medulla, which is where the DNA is extracted from hair. Bigfoots don't have medullas in their hair, interestingly enough. And that complicates the process of getting DNA samples from a hair itself, because the hair doesn't have a medulla. To get the DNA samples, you've got to have some tissue samples, some blood or some other kinds of substances attached to the base of the hair in order to get viable DNA out of that. That to me is just, I don't know, that's interesting. And all the testing that they do on DNA is, again, beyond my head. So, per BFRO, the Sasquatch is covered with hair, not fur, differentiating between hair and fur. Fur has guard hairs and an undercoat, while primate hair consists of one type of hair alone. The Sasquatch, being a primate, does not molt its hair, but is replaced one hair at a time. Hence, it's not found in woolly batches. I don't know if Craig Roach has tuned in, but number one, of course, this comment reflects a blood and flesh and blood bias to it, or view of it. Not everybody believes that Bigfoots are flesh and blood. I'm not sure why they can't be flesh and blood and have paranormal abilities. Humans do. Why not Sasquatches? But so they're full blood, flesh and blood, and that bias is going to come through in, of course, the BFRO's comments, and that's not a criticism. That's just the way it is. If I was reading from somebody who was biased towards woo, I'd be reading their words, and there's always a critic. So what else we got to see? The color of the hair ranges from black, dark, 50% black, dark, through various shades of reddish brown and gray to white. The body can have varicolored patches of hair. Older animals have increasingly gray hair, though the color does not appear to change from childhood to adulthood. The only thing we'll get into that. I don't know if Craig's there. Hey, Craig, here, I see your name. Buddy, let me digress just a second. This line, I'm wondering what you think about this line. The Sasquatch, being a primate, does not molt its hair, but is replaced one hair at a time. Hence, is not found in woolly batches. I don't know if you have any comments you'd like to share about the clump of hair that you had, but from the pictures that I saw of it, it looked like yours was, I don't know if I'd call it a woolly batch, but there was a bunch of hair there, interestingly enough. So that's a striking perspective on Bigfoot hair. Craig is a research partner or friend of mine, and he found some hair. We went for a walk together, me sitting on my couch, and Craig on his cell phone. We took a cell phone hike together back when he was just learning about all this stuff. And he found this huge clump of hair. And I don't know, he sent me about 8 strands, one of which I always wear around my neck. So I don't know, Craig, what you think about if you found a woolly batch of hair or not. Okay, we did that. Hair is variously glossy, clean, and shiny, fluffy, or dirty, matted, and unkempt. Angora goat dreadlocks. Dreadlocks, that's interesting to me. That's a theme that seems to be coming through. Probably a function of native curliness, age, or of recent immersion in the water or lack thereof. That has something to do with the muddy hair, I'm sure. Females have been reported to be cleaner than males. Does that sound familiar to anyone else besides me? That's not what I want. That's not what I want. That's what I want. Craig, coming at you again. Hair length ranges from 3 inches to around 2 feet. 15 inches is the longest measured in hand. I don't know if you've measured the total length of yours. Longer observed in the wild. There is no taper or color band other than graying with age. Long hair covers the head and almost invariably the ears. Very short hair on the face. Occasional reports of heavy hairiness in male faces, a mustache or a beard, versus no facial hair in females. Long hair across the top of the shoulder, once described as a bouncing cape. But fine blonde hair occasionally looks similar to Sasquatch hair. Hence, this is important, I think, hence there is no absolute distinction that can be made with the blonde hair there. Hair from other forest species like rodents, carnivores, ungulates can be differentiated without question. But what's left said in there is what can't be differentiated without question. Under the microscope, the average diameter of the hair is 60. I don't even know what the verbal translation of is. 65 is the average. These values derived from 15 separate collected samples in 4 different states. The cortex has a uniform reddish tinge plus fine pigment granule distribution, whereas the medulla is absent. And that goes back to those pictures that I showed you earlier, guys. Intense efforts at DNA analysis of the hair have been uniformly negative, possibly a function of lacking the medulla. Again, that's where the DNA is stored. Most human hair has medulla, if not only fragmentary. Long hair on the forearms like a spaniel, different orientations of hair on the back, breasts in females hair covered, contrary to a mistaken claim in the literature, long hair on the buttocks, sometimes overhanging them, groin with enough hair to obscure genitalia, and long hair on the calves, like bell-bottom pants. I wore some of those pants back in the day. In a Sasquatch observed standing in the snow. So that's where the bell-bottom pants kind of appearance came in. The hair stood visibly on end in situations where the Sasquatch appeared frightened. I'm reminded of dogs reading that last statement. when they get their haunches up. Hoss, my dog, who's doing great, when he gets his haunches up, if he's frightened or he's agitated. Seeing that in Bigfoot. So let me just take a break there. Any questions, please, if you have any. That's the BFRO view, official view from their website of what a Bigfoot looks like and is. And again, it's a flesh and blood view. There are other people with different views, and I welcome those folks as well. So I've thought or heard of I've heard it called the Ketchum Bigfoot DNA Study. And I actually, for the first time on my own, went and looked at her paper and for the scientific name of the things. So I guess it's in common nomenclature. It's also called the Sasquatch Genome Study. I've heard it called that. But the title of the paper is Novel North American Hominins, Next Generation Sequencing, 3 Whole Genomes and Associated Studies. Melba Ketchum, and then a long list of authors who, if you look them up, it's quite a crew. All seemed very qualified, at least when you go to her website and look at the authors. One thing I'll say, and this was just one insight I noticed in preparing, in her list of authors on her website, Dr. Ketchum, she has pictures of most. Not everybody gave their picture. And then there's one anonymous out of Ontario. I don't know who that might be. If anybody knows who that might be, I'd be interested in hearing about that. But those of you familiar with the history of this paper and some just legend and history and Bigfootdom have probably heard of the Smaja stake. And as best as I can tell on this paper, Justin Smaja isn't listed as an author. And unless he lives in Ontario, Canada, which for all I know he could, I didn't research that deep. He's not listed in the paper or on her website. But we'll talk more about Justin Smaja in a little bit for you guys who may not be familiar. This is kind of my own just quick summary, methodological overview of the study. Multiple independent Bigfoot organizations, and we're going to talk about this, or maybe right now, and come back to it again then if we do, as well as individuals. So it was Dr. Ketchum, and then you'll see some familiar faces on the website associated with the study. But some of them at the time were affiliated with an independent Bigfoot organization, some of which were already doing their own DNA website. Or not website, I'm sorry, their own DNA research. And you mix in the representatives of that organization with some other folks in the study. And that's an interesting mix to me. But they gathered 111 hairs and tissue samples from North America and Bigfoot province. Sorry, guys. They gathered 1,100 hair and tissue samples from North America with Bigfoot provenance. I was trying to add a little French into my show, and Screwed up the word. DNA testing was pursued. Of course, that's the whole point. Final sequencing of the Sasquatch genome. I cut out about seven slides from Dr. Meldrum's stuff between starting the DNA testing and the final sequencing of the genome, because there's a lot of guys. Boy, I'll say it again. If you haven't read genetic studies and talked with people who are geneticists, it's a whole nother language, whole nother field that, as a psychologist, I find, and one of the things I try and do on my show is explain words that might seem common to me as a psychologist, that have just become normal for me, that other people don't know. So if I use a highfalutin word, I hope I catch it and explain it further. Or if I ever use a word, if you want to explain further, by all means, come along and ask. But so the sum total of my DNA understanding as it relates to Bigfoot is you can only get certain kinds of DNA, and if you don't, from what I'm understanding, you'll notice tissue samples. I included the word tissue samples, and I had another slide. I don't think I included it. Maybe, let's see. Let's go for it if I got it. Here we go. Bigfoot hair, blood, mucus, toenail, bark scrapings, saliva, and skin with hair and subcutaneous tissues attached. Ooh, I didn't think about doing this. Let's go back. Subcutaneous tissue. How deep does that go? I'm going to enlarge, guys. I don't have glasses. Where do I do that? Thank you. Subcutaneous. So that's the lowest level of skin. That's not just scratching the surface, the flaky area or the deeper tissue. That's going through the middle of the skin structure and into the subcutaneous tissue. So that's a bit of flesh, I think. I don't know how thick. Like if you think of this as a steak, like a fillet or a loin, a beef loin, Jesus, and you're going to slice kind of diagonally across it, that would be a good chunk of skin. So it's the subcutaneous tissue, again, which you'll notice on the bottom there, is it both innervated and vascularized. It's got a nerve in the subcutaneous tissue, and it's got blood vessels in the subcutaneous tissue. Okay? So that's the type of material they needed in their study to be able to get the DNA extracted from, because they weren't getting it from a Bigfoot hair, be it dark or reddish brown, because there's no medulla, that brown stuff in the center, in a Bigfoot hair. And that brown stuff is where the DNA is typically found. So let's get back. So as you're looking for DNA, it's apparently pretty easy to how I remember this is M for mother. Mitochondrial DNA begins with an M, and mother begins with an M. So the mitochondrial DNA is the mother's DNA. It's 100%, well, it's human, Homo sapien, I guess. Nuclear DNA, and by nuclear we're referring to the nucleus of the cell. That's how Dad makes his contribution. And what the Ketchum study found was that and so it's very difficult, apparently, for reasons I don't understand, but the hair not carrying the DNA being one of those difficulties in getting a good sample. But the nuclear or the dad DNA, nuclear for nucleus, is a mix, if you will. I think the word mosaic, I read, it was used to describe it, of unknown human species, known and unknown human species. So that's where the mix of the unknown species comes in, is in the nuclear DNA. And that's a lot more expensive to run the extra testing on that. It's a more complicated process. And here's the thing. So, they and I should have defined genome. I should have looked it up myself. But the Sasquatch genome, she started with 1,100 hairs, and it came down to, look at that, done on 3 tissue samples. So after she got all her DNA collected by, a nationwide group, who, by the way, I think one of the strengths of her study is the training she outlined and put through for her people submitting samples and how to collect. I mean, it's not like she just said, throw it in a baggie and throw the dirt in there too. She really, as I understand it, as I did my reading, she went through some significant training with her collectors to ensure that the samples were done as sterilely as possible and were collected as uncontaminated conditions as possible. Okay? But I don't understand what I read completely. But in trying to work through the DNA's language, this genome is a combination of multiple step decisions between beginning DNA testing and getting to the point where you somehow, it's magic to me how, but somehow created a genome which is a combination, somehow combined, or it's a genome. It takes a genome from the mother and it takes a genome from the father to be able to create a Sasquatch genome. So there's some chemical stuff going somewhere between starting testing and coming to your point of the genome. And that's part of what I left out of I didn't want to read through that stuff. I didn't understand it at all. But out of 111 hair and tissue samples, 3 were sufficient to be part of that genome. Talk about dropout rate. When you write up a study, here we go, somebody's helping. Troy in Tennessee, buddy, how are you, my friend? Genome, the complete set of genes or genetic material present in a cell or so yeah, mom and dad are both there. Um, and then the debate begins between detractors and supporters. Have I missed any other questions? Thanks, Troy. I appreciate you. Nice seeing you. The debate starts. And so, of course, her conclusion is 3 of the Sasquatch samples were subjected to next generation whole genome sequencing, each of which independently yield high qualities of complete genomes. So they got a good measure, according to Dr. Ketchum, of mom and dad's contribution in this genetic material that they examined. In summary, our data indicates that the Sasquatch has human mitochondrial DNA, but possesses nuclear DNA that is a structural mosaic consisting of human and non-human DNA. So, that's her conclusion. And, objectively, since then, people have drawn lines in the sand. And I think probably I shouldn't even call her Melbourne. I don't know her. I've never spoke with her. Dr. Ketchum's work has been one of the more polarizing, not that it was her intention, but it's one of the more polarizing topics, I think, in the Bigfoot community. And if you really want to get in deep, I think Kevin, I think Kevin is one of these people who know about Kevin Harder. Nice to see you, buddy. You're in this show. You get a plug in this show, my friend. Kevin knows his genetics and can explain it to you guys in the group much better than I could ever hope for. Kevin Harder is in the chat, ladies and gentlemen. So thanks, Kevin, for coming in, buddy. I think I may have just dumped some work on you. I'm sorry if I've done that to you. So, again, I'm not here to pander to people who are fans of Dr. Ketchum's work or the detractors of her work. I'm kind of sharing my own experience. Doesn't reflect Doug Hycheck's view or any of the Untold Radio show. These are my thoughts. But my intentions here is to simply use Dr. Ketchum's study as a teaching tool on certain aspects of the scientific method and scientific research. I'm going to highlight 2 basic issues that I found in investigating her work. First problem being, it was published in a journal bought by the primary investigator of the study, ostensibly to be able to move forward with the work in a timely fashion. So, the first problem, and I don't know if it gets small or big screen for you guys when it does for me, but let's go big screen. So we're looking at the scientific method here. If you're a new viewer to this show, you'll see this coming up a lot in the word scientific method a lot, because what I'm trying to encourage is for people to understand how psychological science can be a bridge between the experiential researchers and the scientific researchers. Psychological science, I typed this on, I forget who I was typing with, But I think I said it well today when I typed that what psychology as a science has done is we've tackled critters like emotions. They're intangible. You can't directly observe them. You can observe the behavioral manifestations of those, but you can't go into a person's heart and tell how they feel or into their brain. Well, if you're in their heart and brain, probably not going to be feeling well, but you can't directly measure that. And what psychology has done and been very successful in doing is taking that intangible thing, an emotion, and being able to measure it. The same with intelligence. You can certainly see the manifestations of a person's intelligence, but you can't put your fingers on it. Here's his intelligence. You can't touch it. You can't feel it. You can't see it in its rawest kernel or seed. You can see the manifestations and the growth and bloom of that seed through that person, but it's not raw intelligence. And so I feel kind of like that makes us good for Bigfoot because, in a lot of ways, many things about Bigfoot are intangible. Many of the woo community questions, for sure, are intangible. And how the hell are you going to measure that? How are you going to measure somebody opening up a dimension in time or space or from one dimension to another? Let's try and replicate that in a scientific experiment. But so I'm pushing the scientific method. And so at the point that we're talking about right now, Melba would have been through the whole process. The scientific method begins with making an observation, then asking a question, testing a hypothesis. Folks, before we jump to a theory, let's start testing hypotheses first. If an idea comes to your head or you have an explanation for something, don't let that become your theory. That's your hypothesis. A theory is developed after a hypothesis has been continually tested and retested and retested and come out with similar results. That's how a hypothesis becomes a theory. But in this one pass method that I've got up on the screen, you form a testable hypothesis. And it's in the forming the testable hypothesis part and making predictions based on that hypothesis. In between those two blue squares is where you're designing your experiment, where you're making decisions about how you're going to set things up, what exact operational variables are you going to assist, what's your independent and dependent variable and all that. And I haven't even, that wasn't even in my advantages in my book. We'll get to that one day. But so when you design your experiment based on what your hypothesis is, because you're trying to prove your hypothesis. I'm sure we understand that. And so you're beginning to design your experiment in that stage and in the next stage where you're making your predictions based on your hypothesis. In fact, you've got to know what those predictions are when you start designing your experiment, because you've got to, in designing your experiment, try to control for all sources of error and bias when you're designing even a Bigfoot experiment. And that's where we need to be going, guys, because if we don't go there, I'll make this last point and then get off my high horse. We're trying to scientifically prove Bigfoot exists as a species. Well, that's my goal. And I can't think of any other reason to prove it other than as a trophy for myself and my ego. But we're trying to scientifically prove Bigfoot exists. And psychologic science has the experimental design and statistical analyses parts of the scientific method that open up the bridge into, I hate to even make the distinction, but up into some of the harder sciences and more theoretically driven sciences and more mathematically driven sciences. But we got to speak the same language as the scientists that are going to be judging whether Bigfoot is a species or not. So I appreciate the experiential, because the experiential work that's been done, um, that forms a foundation for where the science work needs to begin. And that work is done. And I won't go that far. That work is done. We've been doing that for 60 years. It hasn't gotten us anywhere. We need to be taking the next step forward in our scientific ability to prove that Bigfoot exists. If we had been doing this 60 years ago, guys, we'd already be there, or we'd be darn close. So I'm trying to get people to join me in science. That's the overall theme of it. But referring back to now, Dr. Ketchum, she's already went through all these steps down to the point of reiterate or iterate. And you get several swats at the ball when you're doing an experiment. And so if your experiment fails, you're on the right side of that ladder and you see the red arrow. When your experiment fails, you do not pass go. You go back up to the looking at the experimental design levels, and you go back and you look at your hypothesis and see if something needs to be tweaked or improved upon. If your experiment bears out the fruits of your hypothesis, confirming your hypothesis by rejecting the null hypothesis, and we'll get into that in the future, then you begin the publication part. And ultimately, getting published is one of a couple ways to get peer review. Okay? So it was during the time, through circumstances, that so a lot of my source for this book was Scott Carpenter's material that'll be referenced here in just a few slides. And he was a supporter of Dr. Ketchum's, and in that book, where's that book? I don't want to go through all the slides. In that book, he explains certain situations, and one of the things he addresses in the book is why she went and bought the De Nova, I believe is the name of the publishing company. She submitted her work. She's on the green line here. She submitted her work to got other reviewers, got other peer review magazines to look at it. For one reason or another, that didn't work out. And my impression is that the paper was become a challenge to publish. And as the story goes, as I understand it, and I bet you there are better experts out there in this than me, she had submitted the paper. I don't know if the company was called De Nova then or not, but she had submitted the paper to that publishing house. It had gone through a peer review at that publishing house, the reviews of which were favorable to her material. And somehow in the transition between her paper being reviewed, and for some reason this guy wanting to sell the journal, that's when Dr. Ketchum had the fortune or misfortune of purchasing the publishing house. And on the outset, I'm going to say clearly, how do you spell conflict of interest? And I think that's got to be one of the her things from a scientific perspective. I'm sure friends and family were happy for Dr. Ketchum. I would have been excited if I just bought a publishing house, that's for sure. But you know, buying a publishing house, and then the first paper being covered in the publishing house's history, I think, Bigfoot paper in history for sure, is your own work. That spells conflict of interest and is a red flag. As an observer looking, as a quasi-scientific observer looking from the outside in. So she's in the green line. Let's move forward. And that's what I was just talking about. I just gave you that story. Nonetheless, well, one thing I didn't mention in that story that I wanted to, in terms of me trying to understand Dr. Ketchum's perspective through Scott Carpenter's writings, they're in going through the review process, the blind peer review process, with some other not magazines, journals, excuse me, some other journals that she submitted it to. It's a very slow process. Even when everything is successful, it's a long time to submit something and get it reviewed and then tweak either your study or your writing to the criticisms from the peer reviewers. And then you got to resubmit it. And you can go through that step of submission and resubmission. Depending on who your random publishers or reviewers are, you can go through that for a long time, even with a paper that's not about Bigfoot. I know that happened a lot in psychology. But so to me, the red flag of the conflict of interest in publishing in your own journal is pretty glaring. Nonetheless, one of the fundamental and cornerstone tenets for attempting to publish research in a blind peer-reviewed journal is to minimize the development and proliferation of research influenced and biased by the good old boy network or good old girl network, as well as other forms of bias that could impact the blind review peer process. And if I could play some music in the background right now, it would be kind of a merry-go-round kind of music. From my own experience, a blind peer review panel and process is not, it's a great aspirational thing to shoot for, but there's a pragmatic reality to that. I'm going to kind of just spend a few seconds on that. Aspirational goals compared to pragmatic blind peer review realities. The whole purpose of a blind peer review is that the submitting researcher's identity is to remain unknown to scholarly judges and reviewers. They never get to see the person's name or ownership of that paper. And I believe one of the struggles that Dr. Ketchum had to go through was whether to submit it up front as Bigfoot tissue and have people drop out on her, or have to hide that and that not come up until after the hide it or make it not part of the protocol until the results were back. But here are the pragmatic realities of blind peer reviews. A paper starts with the manuscript. And if you're going to be a mover and shaker, you're going to present your papers at national conferences. And big psychological national conferences and other professional fields, the students and studiers of that particular field have meetings and attend talks and that kind of thing. And one of the things that happens is the up and coming scientists and graduate students present their papers at a paper presentation event. And usually that's done throughout the 4 or 5 day event. That'll be there for 4 of those days. Massive room with um, boards up that people go to all kinds of expense and no expense at all except copies to present their papers. And then they have a cocktail hour or two or an hors d'oeuvre hour or two down there in the paper room. And you walk around these long aisles with papers on either side of you, in front, behind you to the left, or in front of you to the left, and in front of you to the right. As you walk down the aisle, everybody's got their papers and their research on whatever they're doing within the field of psychology, in my case. They're presenting their paper, and again, it's over cocktails, over light hors d'oeuvres. But guess who's in that audience? These people who that are peer reviewers are very in the hierarchy of the overall educational system, and I don't know, other larger bodies. These peer reviewers are either big names or somebody at the top of the heap. I went over to Dr. Meldrum's. He's got, if you don't know it, he's got a journal, the only journal that I'm aware of available for Bigfoot research, as well as what I call a thought piece, which is an essay, which Kevin Harder knocked out of the park as far as I'm concerned. But so my point being, people mingle. Researchers talk with researchers who talk with professors who talk with, established drug reps and publishing companies, and everybody mingles with everybody. And so, definitely a social milieu, if you will, develops at a conference. And sometimes it goes further than just a social milieu, but things happen, relationships develop, and politicking is what it boils down to. Whispers between judges about this and that happen over cocktails. Not only do they happen over cocktails, but dinner, and sometimes into the breakfast. The submitting author, again, aspirational goals. We don't know who this guy is. We don't know what this experimental group is, this question, dealing with this question. What are some more of the pragmatic realities? As an established university professor, I'll tell you what happens. I never have been. I'm talking about those that are. Your work gets recognizable. The topic you're studying, and if you're doing successful work in that area, people are going to know you in that area, and your area will take its own signature and style. So you'll be recognizable to a lot of the people that are going to be peer reviewing your work. So that begins, besides sleeping with somebody, that challenges the blindness of any peer review process. And if you don't develop your own style, again, I'm sorry that I can only generalize from what I experienced, which was again psychology training and working. Certain universities, they're going to package their stuff in a different way. They're going to have their own style and signature and reputation that kind of can help people narrow it down. Any questions about it? I'm trying to bring you the dirt behind the peer review process. Not things aren't always as objective as we'd like to think. Sorry. So that whole bias process then can become systematic, and you will find that certain journals are pull for certain topics. You don't want to send your 1 year study on EMG results associated with meditation to the Journal of Clinical Psychology. I don't think. I'm sorry if I offend anyone about that, but uh, you submit your journal to a journal that you think is going to be encouraged to have an interest in your topic. So that further narrows down. The point I'm making about the blindness is a lot of people's work becomes so familiar. Uh, if you study the literature and you're active in the academic literature, you can identify somebody's work just by reading it. So that weakens the whole blindness um, and unbiased aspects. My point is, that's my point. The inner glimpse, the dirt. Um, I think the important point in this slide is even within a prestigious blind peer-reviewed scientific journal, I'm not going to name any because I don't want to get sued, but the biggins, guys, the potential for the blind peer review process, the chances of that not being truly blind or objective, they exist, whether anybody admits it or not. It's a reality. What am I doing? Give you the dirt. Now, in the Ketchum study, this is one of the issues that Scott Carpenter dealt with. Melba's work had already been blind peer reviewed at the point by the journal, at the point that she bought the journal. So why not just publish it as if she never bought the journal and let that be the initial work of the journal? And to get back to another point, I went off on a circumstance on here. Part of what Dr. Ketchum describes in some of her work or interviews I read was that people it felt like it was a conspiracy, that she was people were working against her and wanting, I shouldn't say wanting to keep this work out, but there was a barrier there, and it really blew. I don't want to say blew out of proportion because I don't understand both perspectives, but I think it exists today. People continue to feel really strongly one way or another about this work, but when that red flag of the conflict of interest comes out, um, somebody should have put on the brakes, in my opinion, because it really, this work would have been much better received and scientifically defendable if that buying of that journal at the time, right soon after the peer review happened, we wouldn't be having this. I talked about, I don't know why, it remains unclear to me why this guy suddenly had to sell the journal. Neither of Scott's books explained that very well, and I didn't find anything on Dr. Ketchum's website to what prompted the sale of that. If anybody knows about something about that. But in response to Scott Carpenter, God bless him, great researcher. So he's saying, well, the study was in fact blindly reviewed, so let's just go ahead and publish it. This situation is nonetheless scientifically problematic. Given the already tainted inner workings, the dirt that I gave you, there's always, the process is dragging, and you want to keep things as pure as possible, especially if you're doing research with a living creature. It's difficult to isolate all the extraneous or out of control variables. So I'm pleading with you, or trying to help you understand that basically at the point that she bought that journal, this publication, this paper, was already in trouble. Because even if somebody else bought the publication, it would be the decision of that editor would be to whether go forward with publishing Dr. Ketchum's paper or to not publish it under a new editor. That would have been a choice point by anyone buying that paper or that journal. And again, cast a jaundiced eye. I can't attribute ill motives, although many do. I can't attribute ill motives to her purchasing that journal. It sounds like really bad timing to me. The unfortunate aspect of it is that it also potentially taints and hinders attempts to replicate Dr. Ketchum's work. And as I understand it, she's gathering data again, which is awesome as far as I'm concerned. But at this point, her work's familiar. She's a familiar name. And people feel one way or another about her. It's going to be how is she going to get a blind review on this next bit of research? I guess would be a question I would ask her if she was sitting here in front of me, because now that study is recognizable. How are you going to get around that? And that makes it more of a challenge for the work to be accepted within the scientific community, whether you're a detractor or you're a supporter. That's just a fact. And I just said that. And I said that already. Unfortunately, we wouldn't even have to go into a second issue in terms of professional ethics. And I'm not saying she's unethical. I don't know I believe she's a veterinarian by training. I don't know what their ethical guidelines are, but that would be a big no-no in psychology. And that, just doing that, if everything else came out rosy, in my opinion, the paper still would have been tainted by that unfortunate timing. Here's the one that burns my butt. Not my butt, but okay. The second issue to highlight is one that Mr. Carpenter and Dr. Ketchum feel they have thoroughly addressed and dismissed: the chain of custody of the tissue samples and data. Chain of custody. What does chain of custody mean? In psychology, we called it security. But when you're doing any kind of experiment, I think this would be, um, I think this is true in drug trials as well. The chain of custody of the physical materials, and not only the physical materials, but the computer data within that research. That's your raw data for your experiment, guys. That's the foundation for everything you're doing. So you got to make sure that you maintain responsibility for that. And you got to make sure you maintain security for that. Because if those things aren't done, things can get contaminated, things can get stolen, things can get erased. And so another kind of big red flag for me, and that's why I'm talking about it. You'll understand more in a little bit. Dr. Carpenter, in addressing why chain of custody was no big deal, the relevancy of chain of custody is a fallacy. The DNA is a research paper, not a criminal investigation. Where hair came from and who has handled it is irrelevant when it concerns contamination. Boy, I thought it was just the other way around. I don't know. Um, Again, so now we're talking about to get where are we in the scientific method? Because to complete scientifically complete with it, be whether you like psychology or not, to scientifically prove we got to do Bigfoot. We've got Bigfoot. We got to go through this scientific method hundreds of times and test and retest our hypotheses and find consistent results to be able to then begin to talk about using the word theory. Most of what theories are tossed about are hypotheses that have never been tested within the Bigfoot community. So we got to go through this loop, either one side of the scientific method, hopefully the green side, or the red side of the scientific method. That's part of the scientific reality. Okay. So in further explanation, Scott then quotes Dr. Ketchum. Somebody was told that the lack of chain of custody of our samples neglected our scientific findings. This is not the case. Chain of custody is used in forensics to make sure the unknown samples from the crime scene are tracked from their recovery until they are tested and therefore are not to be altered in any way. It sounds like a pretty good reason to do it to me. I don't know about the rest of you. Keep track of it. If you're doing research, you got to be recording things, be it audio, visual, or notes, and getting details and specifics about the season, the date, the time of year, the weather. Craig Roach could write you a list that I couldn't begin to imagine of. But you've got to be doing something with your records, be they video, audio, or written. That's your responsibility in the research process. That's called science. Okay? So you are ultimately responsible for the chain of custody and or security of your data. Be that data a footcast, be that data a recording, be that data a picture. That's your raw data. You are responsible, and that's carried through in the community, guys. You know, I mean, Craig ultimately found that hair. That's Craig's hair. Somebody found a footprint and cast, a beautiful cast, from where I experienced, which is was either a bluff charge after a tree pushover, or mountain slide, and we never saw any mountain coming down on us. But the next day, somebody went up there and found a footprint, casted it. This thing was just incredible. You could see the individual toe joints. You could see the dermal ridges. The dermal ridges are like your thumbprint, but they're on your foot. So they're your footprint print. And reported Bigfoots have dermal ridges on their prints. If you get good at casting, you can get them. Okay. But to me, or here, she's arguing. She's arguing against her own case. We're not doing a crime. We're not trying to pin somebody down at the scene of a crime. Yes, you are. It's Bigfoot. This ensures that continuing, she says, this ensures that nobody tampers with the sample. The samples themselves are often of unknown origin. However, this testing is a little different than a forensic case in that we are simply trying to determine the source of the samples, human, animal, or unknown. We're not trying to place the individual at the crime of a scene. And I'm sorry, guys, that's what she needed to be doing. The thing about psychological science, we don't directly measure Bigfoot. We're not going to catch them, dissect them. None of that crap. I'm sorry. None of that. We're going to measure Bigfoot phenomena. That's what is accessible to us. That's what we've been doing for 60 years, for goodness sake. Footprints, recordings. Okay? So we're going to use those discoveries, but we're going to get more systematic about how we do that, guard and secure our data, start doing statistical analyses of our data that we can then write up in a paper and be reviewed, peer reviewed. We're going to be doing that hundreds of times if we're going to be successful using any science to prove the existence of Bigfoot. So and in fact, as an experimenter, I want to place, and that's why I started doing Bigfoot expeditions. I want to place that Bigfoot at the scene of the crime. I don't know about the rest of you guys. I want to see one. And don't rub it in, you guys who live with them. So I get incensed in here, I'm not going to go over that. Both the reasons in her explanations basically, in my opinion, beg for the need for a chain of custody and security of the data. But if you recall earlier in our discussion, she was affiliated not only with individuals, but she was affiliated with organizations. And some of those people and some of those organizations already had or were collecting data for their own DNA study. And here chain of custody gets messy, because these are things that people already have owned in some cases and have established their own chain of custody associated with that data and that raw data. And so that is then being submitted as part of the Genome Project study. Well, Craig shared some of those clumps of woolly hair with me, and I guarantee I have them and I value them. And I'm maintaining my own chain of custody for those. And, I'm going to do, you got to take your chain of custody of your data seriously, folks, I guess. But Dr. Ketchum's very words suggest that the main chain, that a chain of custody should be of a paramount issue to such research paper, if for no other reason than to counter human DNA. She wasn't concerned about things getting contaminated by the humans. after she went to great lengths to teach and coach and ensure as much non-contaminated human DNA in the collection process as possible, that chain of custody better be pretty tight. But what happens? She's not concerned about that. And if you meet, I don't know if I should tell this story. Um, I went to see Dr. Meldrum at Cherry Log because I think he's awesome. And I was kind of a twinkly starred researcher at the time. And he did his bare feet presentation, and at the end asked for questions. And I was the first one to shoot my hand up, and I said, "Could you tell me what you consider the strengths and weaknesses of the Ketchum Genome Project?" And I thought I had asked this great question. And he says, God bless him. He says, "Well, I can summarize the good things in just a few words, and we could talk all day about everything that went wrong with it." And my jaw just dropped. There's one of my idols in Bigfooting, and he proceeded to do quite an analysis of the negative aspects of Dr. Ketchum's work. And again, if you want to see that side, and I think it is important to get a balanced view, because I, like most of us, I believe, have a non-DNA perspective on the work, what all that DNA's language means, and what's the significance of it. And I walked away from reading the paper that I'm going to, well, both the papers I'm going to allude to here in a little bit. Um, I walked away from reading those papers like just so confused with all the DNA jargon. And I walked away with an understanding that there seems to be some key progression steps that you've got to go through to reach the point of producing a genome. And somewhere in that, and whether you move on to the next step in that process is based on your last test. And so it's either passes or fails, and you move on to the next step, or you don't. So there's still a lot of interpreting going on. And anytime the human brain gets involved in interpreting, it's not a sin to become biased. We're human bias machines because of our nervous system. Um, um, our perception is contaminated by our life experience from the time we start breathing, and from the time we begin to be able to perceive our world and ourselves separate from that world. What we're perceiving is a biased chain of events. It's not the same reality that other people feel, see, or um, not preserve, perceive. You know, how transmutable is reality? That's another. Um, so, you know, detailed log showing who collected, handled, transferred, or analyzed the evidence during the investigation. In short, it refers to the raw data security. I've harped on that. I'm not going to belabor this. So she needed to tighten up. Think twice about the obvious conflict of interest of buying that. I guess my two main points are at buying that journal at the same time as uh, she was being, or just got through reviewing. I don't know how much, how long, anything like that. but the timing of that was not good. It was unfortunate. Just turned towards the whole the whole scientific community is supposed to be idealists, and I meet a lot of different people with a lot of different ideals in my life, but science has its own ideal. And what I called a red flag, or I called a conflict of interest, that may or may not be part of certain scientific codes that the organization maintains. And, this was physical material. My main point about it is, so the complexity came that she was not only dealing with individuals, she was dealing with organizations and people that already claimed custody, if you will, of the tissue. So people didn't want to send, understandably, people didn't want to send all their tissue to her. So they sent samples of that tissue. The organizations and the individuals that wanted to hold on to their tissue got to hold on to their tissue. And one of the guys that got to hold on to his tissue was a guy by the name of Justin Smaja. And Mr. Smaja, as the story goes, was out bear hunting, apparently out of season, and saw a Bigfoot mother and father, definitely Bigfoot mother, and I think 2 infants. And one of the infants got snatched up. He shot the remaining adult that hadn't gotten over the little hill, but they just kept on moving with one kid, and he shot the remaining infant. And he said when he walked up and picked that carcass up and looked at the face of that infant Bigfoot, that he saw the humanity in it, which I can only imagine. He saw the humanity in it, and he realized, like, one, I shouldn't have done that. Secondly, what the hell do I do now? So he got panicked. He buried some poorly. I don't know how well he concealed. He stashed the carcass. Came back down to town. Talked, I guess. I don't know. Long story short, he went back to get the carcass. And through all this process, came to the attention of news media, Bigfoot researchers, and of course, some of the larger groups from which they had submitted one of those 111 tissue samples. So the thought that maybe Justin Smaja should be charged with murder came up. He started getting pretty anxious, understandably so. I would get pretty anxious about that myself. But fortunately, because Dr. Ketchum didn't maintain 100% custody of all the DNA that she collected, she got people to dole out some of their DNA to her. And if she's already doing her own research, she doesn't. She's a competent researcher. She doesn't need me to be telling her. But in my opinion, in any research I've ever been involved with, there was a person that was designated for all of the data. We didn't just get half of Justin Smaja's steak. It was called a steak because when he went back and got it, there still was some flesh and blood and skin on it. And if I'm not mistaken, of the 3 samples, I believe his was one of the samples that yielded DNA evidence. Scott Carpenter certainly talks about that in both of his books, but I did see no discussion of that on Dr. Ketchum's website. And Justin Smaja's name was nowhere on that site to be seen. That would be the best way to describe it. Justin Smaja's name was nowhere on her site to be seen as a contributor to one of the three samples, I believe, um, that was found DNA, both nuclear, and again, remember the mitochondrial DNA is easy to get. The nuclear DNA is the tough one to find, and that's why we went from 111 down to 3. That's not the only reason why, but a big part of why. She ran into trouble getting people to give her a review or an analysis if they found out it was Bigfoot. I mean, it's not like this woman just immediately went out and said, I'm going to go buy me a company to publish me my research. It didn't work that way at all. I know that is my objective impression, as objective as I can be. I think it's ridiculous to suggest that chain of custody and security when you're handling the physical material and the need for it is a fallacy, because it's not a forensic consideration. It is a forensic research situation. Do not consider these factors or maintain the chain of custody would have and should have been unfathomable, given the potential and anticipated hope for, conclusions that are going to change the face of this earth and what we believe in God. And God only knows if, what's going to happen with the forest. And if Bigfoot gets proven, we're going to feel ripple effects for a bit of time. Oh boy, I'm almost out. I'm not suggesting Dr. Ketchum is being dishonest. I think, again, taking the high road, I'm not going to get into the controversies, but, I think her explanations are within a relevant context of defending her work. And that's part of the peer review process. That's also part of the scientific process. When your paper gets rejected that first time, that's not the end of it. If you give up there, you'll never get published. You got to adjust your work, tweak your research, and submit it again and again until it gets published. And she wasn't having any luck doing that. The results. Unidentified species, we've talked enough about that. Human hybrid. This is a big controversial point for Dr. Meldrum, because humans supposedly can't hybridize. I can't go out and find me a female ape and produce a child that will survive. Gene splicing. Everybody knows about the human hybrid method or issue. Did you know that she found, I think it was a 3-strand DNA spliced into between 2 strands DNA? Kevin Harder, is that the case, buddy? You would be the expert on that. And, more recently, people are getting a lot of traction out of using eDNA, environmental DNA. The problem with environmental DNA, from my perspective, is you can only get mitochondrial DNA. You can only get the DNA material of the mother. And we need to be getting daddy's DNA if we're going to get it. Alternate explanation, Kevin, boy, I wish I had more time. Does mitochondrial introgressions explain Sasquatch Genome Project better than the human hybrid of recent? Kevin, you are the man. That paper impressed me so. All I'm going to say, which is a weak, terrible understanding, is that somehow is it mitochondrial or nuclear DNA? I'm sorry, I forget which, can migrate between species over thousands and thousands of years, with them having only minimal to little contact with each other through, I don't know what kind of process. That's the process I don't understand, is the introgression, the mitochondrial introgression. That sounds like a very complicated process, but interestingly enough, it would explain the results of the Ketchum study without hybridizing, without a female human getting raped and by an unknown species. Carpenter's books, I enjoy them. I after the first time I looked at his Nephilim Among Us Revisited, I looked at his video plates and his captures in that book, and I was afraid to go back into the forest by myself. I've only been back into the forest once, and that was early on when I started Sasquatching. Before I saw that book, the photo plates in that book, I was fearless in the forest. And when I saw what he was catching on video by pointing his camera beside him, guys, that convinced me otherwise. And one more mention. Oh God, I don't want to jump in on my neighbor. Here we go. I'm gonna have to wrap up quick, folks. I just want to give a heads up. God, I wish I could remember the guy that I spoke with. I spoke with somebody who spoke DNA through a thread. I'm sorry, sir, I can't remember your number, but your name is vaguely familiar because I think you're one of the OGs, or close to it. But look up John Timmer. I think what this controversy boils down to is the geneticist's understanding of Dr. Ketchum's work versus us mere humans understanding of those decision points. Earlier, I mentioned that it takes a lot of it's a multi-step process, and there's a lot of theoretical and hypothetical situations addressed. And people read the same thing. Perception, guys. It's about perception. People perceive the same thing in different ways. And that's a good thing in the scientific community, because when we get it right, then we got it right. And that's why a blind review peer process is so important. Thank you very much for attending my show. Keep your minds open. Keep your boots on the ground, and keep your heart with your highest power. I wish you guys well. Good night.